Hey everyone, welcome back to my channel Nityashree's Bio. Today we are going to get into a new topic, that is biotechnology principles and processes. Let us begin the chapter with small introductory part. So, what is biotechnology? Biotechnology deals with techniques using living organisms or enzymes from them to produce products and processes useful to improve human health and society. Here, main motive is to support the human health and society. So, we use certain techniques and technology in laboratory conditions, and we use living organisms or the enzymes which we obtain from them to get such products. For example, production of fermented products and production of vaccine. Next, we will move on to the definition that was given by European Federation of Biotechnology. This is a non-profitable organization which helps in safe and sustainable use of biotechnology. So let us see what do they say. So, the definition given by European Federation of Biotechnology EFB is: biotechnology is the integration of natural science and organisms, cells, parts thereof and molecular analogues for products and services. It means to tell that it is combination of natural science, organisms, cells and their parts and molecular analogues. Molecular analogues are nothing but the compounds which looks like a natural ones for the products and services. Come on, now let's move on to principles of biotechnology. There are two main principles. One is genetic engineering and second one is chemical engineering. Genetic engineering, altering the nature of genetic material, DNA and RNA, and introduce these into host and results in change of phenotype of the host. What does this mean? Altering the nature of genetic material. Here we are going to change the nature of genetic material, DNA and RNA, and introduce these into host. Host is an organism which is going to accept the change, or which is going to accept the DNA which has been altered and results in change of phenotype of the host. When we introduce an alien piece of DNA into the host, it will start expressing that particular character, thus changing the phenotype of the host. Next principle that we have is chemical engineering, maintenance of sterile microbial contamination free condition. Sterile condition is nothing but the room wherever we prepare these particular things will be very neat and clean. It will be contamination free. It will be free from germs, viruses and other microbes which we do not require, to enable growth of only the desired cell or microbes. Here we can grow whichever cell we want, or cell of interest or microbes of our interest without any other contamination. In large quantities, you can start growing them in very large vessels in very large quantities for the manufacture of various enzymes, vaccines, antibiotics, etc. Now we will be focusing on the principles of genetic engineering. Burke is considered as father of genetic engineering and was awarded Nobel Prize in the year 1980 for creating first rDNA molecule. Conceptual development of the principles of genetic engineering includes. So these are the three basic steps in genetically modifying an organism. First one is creation of recombinant DNA. Second one, use of gene cloning method. Third one, gene transfer. So we will be studying what each one of them is in upcoming sessions. These techniques help us to isolate and introduce only one or set of desirable genes into the host. So in a genome, we will be having certain regions which we want and certain regions which we do not want, which we call as desirable and undesirable genes respectively. So we can get only the desirable genes using these three techniques and introduce into the host for changing its character.